Hello friends, and welcome to this video lecture on topic C3.2 Defense against disease. Our guiding questions are: how do body systems recognize pathogens and fight infection, and what factors influence the incidence of disease in a population? So many objectives for today. We're going to define pathogen. We're going to talk about how skin and mucous membranes are the first lines of defense against disease. We're going to talk about the clotting cascade when that skin gets broken. We're going to distinguish between innate and adaptive immunity. One of our pieces of innate immunity is phagocytosis with macrophages. They're going to present some antigens. We're going to define what an antigen is. We're going to talk then about adaptive immunity, T cells and B cells, antibodies that are produced by some special kinds of B cells. We're going to talk about blood type and how it relates to antigens and antibodies. We're going to describe HIV transmission, the effects of HIV infection on the immune system. We're going to talk about mechanisms of antibiotic activity. We're going to define zoonotic diseases and then describe how vaccinations can lead to immunity. We're going to wrap up with a little bit of math. So if you want to pause this and grab a calculator, you're going to want it at the end of this lecture. Pathogens are disease-causing agents. Pathogens can be any one of many kinds of organisms. They could be protists, they could be bacteria, they could be fungi. Of course, viruses can also be pathogens, but they are not considered living. We have learned a lot about pathogens over the last couple hundred years. This sketch is showing children lining up at a well to get water. In London and all over the world in the mid-1800s, people would collect their human waste and just dump it into the streets. It would wash into the water, and then these kids who were bathing and drinking this water would also be bathing and drinking poo, basically. And then there was a cholera outbreak. One doctor noticed the connection between this well and the number of infections that were popping up, people that used that well. And so he shut down the well, got the city of London to shut down this well, and then the incidence of cholera decreased. So he was able to figure out that the pathogen was originating at that well and then slow down the spread of infection by turning off that source of infection. Another example of some doctors and doctors being observant is here. This is showing Dr. Ignaz Semmelweis of Austria, who worked in a maternity ward, and he noticed that the women in childbirth who were treated by doctors died far more frequently than did women who were helped through childbirth by midwives. He thought that perhaps the doctors who would do an autopsy and not wash their hands after touching dead bodies go straight into the maternity ward to help women have babies. Perhaps something was being carried on the hands of the doctors from the dead bodies to the women, which was leading to that increased mortality of women post-childbirth, because the midwives washed their hands and women were not dying as frequently. So Dr. Semmelweis said, doctors, wash your hands after you touch a dead body before you touch a live one, and voila, the incidence of women dying post-childbirth decreased because doctors washed their hands. So let's talk about some of the ways that our bodies have to fight off these infections. The first line of defense is skin and mucous membranes. The skin has a couple different layers. We have the epidermis more toward the outside. The dermis is just under it. The epidermis is mostly dead cells, and these set up a physical barrier, preventing pathogens from being able to get into our bodies. Just under the epidermis is the dermis, which is very much alive. Lots of sweat glands and capillaries, nerve cells. We're going to talk about some of these cytokines and T cells and macrophages in the next couple of slides. So a lot of chemical barriers also to infection in that dermis. As the epidermis is sloughed off, these dead cells just fall off our bodies all the time. The dermis cells will replace them, so we have a constant supply of skin cells, creating that physical barrier. Kind of fun, we also have some helpful bacteria that live on our skin, help to prevent the bad bacteria from getting to us. The other component of our first line of defense against disease are the mucous membranes. We're not going to worry too much about all of these details. Again, B cells and T cells, macrophages, we're going to talk about those more coming up. What I want us to focus on is just this layer of mucous membrane. So we have these cells that are secreting lots of mucus. The mucus is a sticky substance. The pathogens that might be trying to get into our bodies literally can get stuck in that mucus, and then the cilia on these cells, the epithelial layers of the mucous membrane, can kind of wave, and then they can push that mucus with all the stuff stuck in it up and out of the body. We have these mucous membranes where we don't have skin, so inside our trachea, which is the tube that leads to the lungs, inside our sinuses, inside our nasal passages, also inside our urethra. This is the tube that takes urine from the bladder to outside of the body. And then also in the vagina, there are quite a few mucous membranes to prevent bacteria from getting into the body through the vagina. Sometimes the skin and those mucous membranes can be broken, and in these instances we have this amazing clotting cascade to help to close up the wound before too much blood gets out and before too many pathogens can get in. Cascade is a series of reactions where one reaction triggers the next, This clotting cascade leads to the formation of, you guessed it, blood clots. In our plasma is the watery basis of our blood, we have some proteins. Those proteins are floating around all the time in the plasma. We've got some prothrombin, we've got some fibrinogen in the plasma all the time. Also in the plasma, we have some platelets. Platelets are fragments of blood cells that help to also trigger the clotting cascade. When there is a wound, the wound is going to release some clotting factors. Those clotting factors, in addition to the platelets, are going to catalyze the conversion of prothrombin into thrombin. That thrombin is the active version of this enzyme that is then going to catalyze the conversion of fibrinogen into fibrin. Fibrinogen is soluble, floats around the blood, easy peasy. Fibrin is an insoluble fibrous protein which creates this net at the site of the wound. That's going to catch red blood cells and white blood cells and all kinds of other things, which is going to prevent too many more blood cells from getting out and also prevent pathogens from getting in. We're going to talk about two main categories of our immune system. We have both innate immunity and adaptive immunity. Innate means born with. This is the immune response that we have from birth, and it is non-specific. So anything that does not belong in our body, our innate immune system is going to go and attack. We have lovely proteins on our cells that let our immune system know what cells are us, those cells that are self, versus what cells are non-self. Our phagocytes, which are very large white blood cells that are going to kind of travel around, do some endocytosis of pathogens, gobble them up into some vesicles. These are parts of our innate immunity. Those macrophages are going to phagocytize or complete endocytosis on any old pathogen that we don't want to have in the body. That's different from the adaptive immune system, which is going to work only against specific pathogens. Here is a slightly more detailed look at phagocytosis, this endocytosis of a pathogen. So here we've got this pathogen, perhaps it's a virus, and it's got those proteins of its capsid. The phagocyte, this white blood cell, sometimes we also call these macrophages, macrophage, phagocyte, same thing. These guys are going to notice that these proteins on this pathogen do not belong. And so there's going to be some endocytosis of that pathogen. That pathogen is going to get put into a vesicle. There is going to be a merging of a lysosome that contains digestive enzymes called lysozymes. Those digestive enzymes are going to break down the pathogen in the vesicle inside the phagocyte, the macrophage. And then what's crazy cool the macrophage is going to choose a piece of the pathogen to be what we call the antigen. The antigen is the specific piece of a pathogen that is going to trigger our immune responses. That antigen that is chosen after digestion of the pathogen is going to be expressed on the cell membrane of this macrophage, which is now considered a dendritic cell or an antigen presenting cell. This guy is now specific, no longer innate. And here we're going to look at a few more details of that specific or adaptive immune response that we have. Antigen is the piece of a pathogen that is going to trigger an immune response. It is frequently a protein that was found either on a cell membrane, or if we're talking about a virus, then in that capsid protein coat. Again, our macrophage is going to phagocytize or do some endocytosis to gobble up a pathogen. It's going to digest it. It's going to choose a piece of the pathogen to present. Here we have that presentation of the antigen. The first cell that we are going to have engage in this adaptive immunity is going to be our T cell. So the T cell is going to bind to that antigen that is being presented by the phagocyte, and it is then going to go on to stimulate the next cell in our in adaptive immunity, which is the B cell. We're going to talk a lot about T cells, and then we're going to talk a lot about B cells. So get excited for all of these details. Again, these T lymphocytes, or T cells for short, these T lymphocytes are activated by our macrophages. Here are those antigens. Our CD4 plus T cells are known as helper T cells. That's just the name of a protein that is on these T cells that makes them helpers. They are activated by specific antigens on those macrophages. They're then going to display the antigens on their own cell membranes. They're going to release some cytokines. Cytokines are specific types of ligands involved in chemical signaling that is then going to help to activate the next guy that we're going to talk about, those B cells. Some of our T cells will be memory cells and will remember what these specific antigens look like so that we can stimulate an immune response really, really fast in a secondary infection. We have other kinds of T cells that are not helper T cells. We also have cytotoxic or killer T cells. They have a different protein, CD8 instead of CD4. These guys release chemicals that kill the pathogens. So not just activate other things, but instead go out and literally kill the pathogens. These T cells are also activated by our helper T cells. So we have macrophage activates helper T cell activates all of the other cells. It does that with those cytokines. Here are examples of cytokines. That IL stands for interleukin. It is just a specific type of cytokine. Remember that those helper T cells are going to activate B cells. A specific helper T cell activates a specific B cell. The helper T cell doesn't activate any old B cell. It's going to activate the B cell that is specific for that exact antigen so that we can fight off that specific infection. So this helper T cell is going to activate the specific B cell, none of those guys because they're not a match. These B cells are going to go through lots and lots and lots of mitosis. We call this clonal selection because that mitosis results in genetically identical daughter cells, hence clones. Clonal selection, so many B cells are activated and then clonally selected. Some of those cells are plasma cells. Plasma cells are specialized B cells that produce and secrete antibodies. And then we also have some B cells that are memory cells. We're going to look at those in a couple more slides. These super cool antibodies are proteins. They are polypeptides that are, again, produced by those plasma cells. Plasma cells are specialized B cells. Those B cells are activated by the T cells, the helper T cells. Those helper T cells are activated by the macrophages. Good. So antibodies are pretty cool. They are composed of 1, 2, 3, 4 separate polypeptide chains. And what's kind of cool is we have some that are a little bit longer. We call those the heavy chains, and then the shorter ones are the light chains. These parts of the antibody are exactly the same. This is our constant region. So from one antibody to the next, those chunks look exactly the same. But then we also have these variable regions, and those variable regions are specific to different antigens. So this variable region is going to bind to only this antigen because it's very specific. This chunk of these antibodies is only going to bind to this specific antigen. Again, the rest of it is the same from one antibody to the next. But that specificity, again, allows us to attack antigens and the pathogens that they belong to with a great amount of specificity. What do these antibodies even do? Well, they might bind to the pathogens and get them to clump. And when they're all clumped up like that, they settle or precipitate out of the blood so that it cannot be transported around the body anymore. They also might lead to some agglutination, which is going to link cells together, more clumping or clotting. It might neutralize something, especially if we're talking about something that has a lot of toxins in it. It can also trigger histamine release, which is going to increase inflammation, which sounds bad, but it does actually help to enhance our immune system activity. It can also activate complement, which is a special kind of protein that literally drills holes into pathogenic cells, and then the cells kind of leak to death. Will lyse, all the cytoplasm will just ooze out. That cell lysis is happening here. We can see we've got all that cytoplasm oozing out. And then we also, when we're looking at this agglutination, neutralization, all of these, once we clump these pathogens together, it's way easier for our macrophages to come in and gobble them up all at once. Again, those antibodies are produced and secreted by plasma cells. Plasma cells are specialized B cells. B cells are activated by helper T cells. Specific helper T cell, specific B cell. Lots of mitosis, which we're going to call clonal selection, because we have so many copies of this particular kind of cell. We selected just the cells that are good for this infection. Through lots of mitosis, lots of plasma cells, we're also going to produce some memory cells. These memory cells are longer lived than are those plasma cells. They hang out in our bloodstream or in the bone marrow for a really, really, really long time. But the delightful thing about these memory cells is if I am infected by that same pathogen, that same antigen a second time, these memory B cells are very rapidly activated, differentiate into plasma cells. Those plasma cells pump out, oh my gosh, so many more antibodies than in that first infection. And so we're able to actually kill off this infection far more quickly the second time around than in the first time. Often we have these secondary infections and we don't even know that we're sick because our immune system is so fast to knock out those illnesses we don't even feel symptoms. Pretty amazing. How we test for blood type also depends on those antigens that are parts of our blood cells and the antibodies that we might produce against those different glycoproteins on red blood cells. So the antigen on a red blood cell is going to be that A glycoprotein or that B glycoprotein. People who have type AB blood have both A and B. People who have type O blood have neither the A nor the B glycoprotein. Antibodies are formed for the antigens, those glycoproteins that you don't have. So someone with type AB blood, both A and B are normal. They're considered self. And so there are no antibodies to attack A or B because both A and B are good. They're me. If I'm type B blood, then type A is not me. And so we'll make antibodies that are anti-A, but we won't make anti-B antibodies because B is normal. Those of us with type O blood, which is the most common type of blood, have neither A nor B. So both A and B glycoproteins are bad. They're not us. And so we'll make both anti-A antibodies and anti-B antibodies. People with type O blood are considered the universal donors. Their blood can be given to anyone, but they can only receive type O blood. And if we want to throw in the Rh factor, this is the rhesus protein, a whole other protein that can be expressed on those red blood cells. Most of us are positive. We do have that Rh factor. Some people are negative. They do not have that protein. People who are positive can get blood that is positive or negative. People who are negative can only get blood that is negative. HIV, human immunodeficiency virus, is a virus that infects humans. How it is transmitted from one human to the next is through unprotected sexual contact, through sharing hypodermic needles, and then also from mother to baby during pregnancy, childbirth, or while breastfeeding. You cannot contract HIV through saliva or sweat or tears. You cannot get it from swimming in the same water as someone who is HIV positive. HIV, this virus, is what we call a retrovirus. It's kind of cool because it's got RNA on the inside of it. These proteins on the envelope of the virus bind to helper T cells. And that is really bad and is why HIV and AIDS is such a difficult disease for us to treat. Remember that the job of the helper T cell, after it's activated by a macrophage, it's going to activate those cytotoxic or killer T cells. It's also going to activate the B cells, which will differentiate into plasma cells and memory cells. What HIV does is it binds to, gets inserted into, and takes over helper T cells, eventually killing them. And so we can see that as the number of viruses increase, the number of helper T cells as well as cytotoxic T cells decreases. What's happening is those helper T cells are getting killed off. That's really bad because it means that we're not activating the killer T cells. We're not activating any of the B cells. So we're not making any antibodies and we don't have any memory cells being formed. This is really bad because even just a common cold can kill a person with AIDS because there is no turning on of that adaptive immune response. Luckily, we do have some medicines that we can take to help our immune system fight off some infections. Unfortunately, medicines like antibiotics that we derived originally from fungi, like penicillin, have no effect against viruses. So we cannot use antibiotics to treat viral infections like HIV. We can use antibiotics to fight bacterial infections because the mode of action of our antibiotics is against some piece of a prokaryotic or bacterial metabolic process. Some antibiotics might inhibit the synthesis of new cell walls, or synthesis of cell membranes might be disrupted, or we might inhibit protein synthesis, or we might inhibit RNA synthesis. Lots of ways that we can inhibit, slow down, prevent metabolism of bacteria. This doesn't work against viruses because viruses don't have their own metabolism, and so there is no effect against viruses by our antibiotics. Unfortunately, sometimes we overuse our antibiotics and that leads to some increased antibiotic resistance. Antibiotic resistance occurs when bacteria have mutations that allow them to not be killed off by the antibiotic. So we take some antibiotic and it kills off the bacteria that don't have the mutation, but then the bacteria that do have the mutation, if we don't take all of our medicine, are able to survive and have lots of babies. And all of those babies, okay, they're not babies, they go through binary fission, they make more bacteria cells. All of those resulting bacteria cells are now also resistant. And so the increase in the population of the bacteria that is resistant is problematic. How do we reduce antibiotic resistance besides finishing all the antibiotics that you are prescribed? We only prescribe antibiotics when needed. Don't ask for antibiotics if you have a viral infection. We're going to try to not add too many antibiotics to farm feed. We don't need to give antibiotics to every chicken, every cow, every pig on the farm. If we let them spread out a little bit more, if we don't have our animals walking through their own feces all the time, then we don't really need to add antibiotics because they won't be getting as many infections. Make sure that you're getting vaccinated. The more vaccinations we have, the fewer times we get sick, the less often we need antibiotics. And always, friends, wash your hands. Many infections are species specific. Viruses especially bind to specific proteins on specific host cells. And so lots of diseases, the common cold, only makes us humans sick. However, there are some infections known as zoonotic diseases that can pass from one species over to us humans. An example is rabies. We can get rabies from rabid animals from bites. Dogs and bats, coyotes, other animals could carry rabies, and if we get bitten by one of those animals, then we could also get rabies. How do we prevent it? Don't mess around with animals that you don't know. Make sure that your own pets have their rabies vaccinations. If you do get bit, wash your hand, lots of soap, and then head to the hospital. Another example of a zoonotic disease is tuberculosis. So some tuberculosis can be passed from cattle or other wildlife to us humans, especially in food. Another example is Japanese encephalitis. This is naturally occurring between mosquitoes and birds, but sometimes those mosquitoes can also pass the encephalitis on to pigs or horses, and then also to us humans. And of course, COVID-19 is a zoonotic disease. We know that it was passed to humans from an animal reservoir. We just don't know what exactly that animal reservoir is yet, as of March 2024. Friends, make sure you get your vaccines. How do they work? Why do we want them? Basically, in our traditional vaccines, there are weakened or dead bacteria. Sometimes we have fragments of viruses. That's injected into the body. Basically, it's the antigens without the pathogens. Remember, the antigens are the pieces of the pathogens that trigger an immune response. The whole pathogen is what causes the disease. The antigen by itself does not, but the antigen by itself will lead to an immune response. And so the body will respond to those antigens. Macrophages are going to gobble them up and then present them to helper T cells. Helper T cells are going to activate the B cells. B cells are going to differentiate into plasma cells and the oh so important memory cells. Remember the graph that we looked at a little bit ago, where we have in our primary or initial immune response a little bit of antibody being made. But then in our secondary response, because of those memory cells, we have this crazy fast production of antibodies, big and fast, and we knock out those diseases before we even realize that we're sick. The vaccine can give us that primary immune response. And then when we are exposed to the pathogen for real, we have a really fast response. We knock out that pathogen before we even feel sick. In addition to the traditional vaccines, we now have mRNA vaccines. How this works, scientists decoded the genetic makeup of spike proteins. This one happens to be on coronavirus. We take that genetic makeup, turn it into a piece of RNA. We put it into a lipid nanoparticle. Because it's a lipid, it's going to merge with the lipid bilayer of our cells, and we're going to insert that piece of messenger RNA into our cells. That messenger RNA is then translated by our ribosomes to make spike proteins, but just the spike protein, not the whole entire virus, just the spike protein. Those spike proteins are then expressed on the cell, and then we have an immune response. Our body's like, oh, what's that? Let's attack it. And then we can make some antibodies that fight off the spike proteins that we made for funsies, but also the spike protein on the actual coronavirus should we be exposed to it. Getting our vaccines allows us to build herd immunity. If we don't have very many people vaccinated in a population, then it's really easy for diseases to spread really fast. But if most of us are immune through either a prior exposure or through a vaccination, then the spread of disease is much, much slower. We love to analyze the data that comes from diseases. And so we're going to do just a little bit of practice math here, looking at ways that we can evaluate data coming from pandemics. Two main and easily confused formulae that you want to memorize are percentage difference and percentage change. Percentage difference is a lot to think about. We're going to find the difference between two values. Oh my gosh, I spelled between wrong. Sorry. The difference between two values. So basically just a subtraction. And then we're also going to find the average of those values multiplied by 100 to turn it into a percent. Let's say that I want to find the percentage difference between cases of COVID-19 in North America and cases of COVID-19 in South America. I'm going to find the difference between these two. That's going to be 30 minus 24. I'm going to find the average of those two. 30 plus 24 is 54. Divide it by 2 to find the average. Multiply by 100 to turn that into a percent. 30 minus 24 is 6. 54 divided by 2 is 27 times 100 to turn it into a percent. Handy dandy calculator, 6 divided by 27 is 0.22222, so many twos, times 100 is going to be a 22% difference between cases of COVID-19 in North America and in South America. We also have percentage change formula. This is usually looking at a change over time. For example, let's say that I wanted to find the percentage change in vaccination in rural America in April of 2021, and then in rural America in January of 2022. So I would find my final value, that's going to be 59, minus my initial value is 39, divided by the initial value, and then times 100. Again, handy dandy calculator time, 59 minus 39 is 20, divided by 39 times 100 is going to be 0.5128, so many digits, I'm going to round that to 51%. So there's a 51% change in the numbers of vaccinations in rural America between April 21 and January 22. And on that note, my friends, we did it. We achieved all of these objectives. There are so many. Pathogens are disease-causing agents. Skin and mucous membranes are the first line of defense against disease. But if they break, we've got that clotting cascade where we have platelets and clotting factors turn the prothrombin into thrombin, which turns the fibrinogen into fibrin, which makes a mesh that closes up the wound, makes a scab or a clot. We talked about innate immunity that we are born with, and it is non-specific. Adaptive immunity develops over our lifetimes, and it is specific to the pathogens that we've been exposed to. Macrophages are going to be part of our innate immunity. They gobble up all kinds of pathogens. They are going to digest those pathogens and then choose a piece of the pathogen to be the antigen. The antigen will be the piece of the pathogen that triggers an immune response. We've got T cells, we've got B cells. We have some super cool T cells that are helper T cells that activate the B cells. B cells can differentiate into either plasma cells that make lots of antibodies or memory cells that help us stay immune over long periods of time. Those antibodies will stick to specific antigens and help us kill off those pathogens a little bit more quickly. Our blood type also depends on antigens and antibodies, and I have type O blood, so please do not give me a transfusion of type AB blood. It will not go well for me. We talked about HIV transmission through body fluids. The effects of HIV infection is the reduction in the helper T cells, which means that we cannot activate the killer T cells or the B cells. Antibiotics are often derived from fungus, like penicillin, and they act only against the metabolism of prokaryotes. So they work against bacteria, not viruses. Zoonotic diseases are those that have jumped species, so from an animal to us humans. Vaccines can give an initial or primary immune response without us getting the disease, and then when we're exposed to the actual pathogen later, we have that crazy fast secondary response. And then we did some math: percentage difference, percentage change, percentage difference. We're going to find the difference of two values divided by the average of those two values times 100. Percentage change is going to be the final value minus the initial value divided by the initial value also times 100. Great work today, my friends.